Does anybody have any questions while we're waiting for the top of the hour? One of these days you're going to just astound me by having any questions. Did everybody get the email I sent out about the quiz material for Monday? Okay. This is going to be a little bit unusual because this is information that I'm quizzing you on right now because it's at the end of the chapter we're reading. But we're really not going to use the information until a little bit later in the semester when we talk about cancer and cell signaling. It's a big part of understanding what's going on in cancer. So this material will be on the quiz, obviously, but it won't be on your first exam. Okay? So it's kind of a preparation for down the road. Please, as I said in the email, pay special attention to the chapter summary at the end of the chapter and then also the diagrams for that section. There's a real tendency in reading a textbook to skip the diagrams, and that's the last thing you should do. Because if you can understand and reason through those diagrams, you know what's going on. Okay? So please make sure you pay attention to those. They're really packed with information, and that's the kind of information that would be good to have on the notes you can use on the quiz. We do have an exam coming up not next Tuesday, but the following Tuesday. I say that because it's not too early to begin studying for your exam. And one of the first things you can start to do studying for your exam, if you haven't been doing anything, is start reviewing your old PowerPoints and notes and use those PowerPoints and notes to answer the study guide questions that are posted on the web. The study guide questions typically will follow the order of the material in the lecture notes and the PowerPoints, and you should be able to answer them with that information. I want you to read the book as background and reference material. But for an exam, I will not pull information out of the textbook that we have not talked about in class. So your priority in terms of studying for the exam should be your notes. And the study guide asks you to write out answers based upon those particular materials. It's a good idea to have someone look at your answers. And that would be a very good thing to do in one of the mentoring sessions is if you write out answers to those study guide questions, then have Austin take a look at them. Because it doesn't help you at all to write out an answer if it's a bad answer and then reproduce that answer on an exam. So get some feedback. It has not been uncommon for those study guide questions to appear as short answer or short essay questions on the exam. So if you haven't been studying, those are the things that I would urge you to begin working on. Don't wait until the night before. Okay? Too much material. Okay. So if we have this particular situation of a hypothetical cell, which is really just a bag, a piece of dialysis tubing, filled with these particular solutions, 0.03 molar sucrose, 0.02 molar glucose, placed in an environment, which is a liquid in the beaker, which contains this amount of sucrose, glucose, and fructose. What's going to happen to the cell as a result of osmosis? Is it going to get bigger? Is it going to stay the same, or is it going to get smaller? What's going to determine that? The solute concentration of the cell compared to the solute concentration of the environment. Where is the total solute concentration greater? Inside the cell, right? And what do solutes or salt do? They suck water. Okay? So water is going to tend to move from an environment that is hypotonic, less solute, to an environment that is hypertonic, that has more solute. Okay? And eventually, the cell could rupture and burst. Now, there's an assumption that we make when we do this analysis, and that is we have to assume that this membrane is impermeable to these particular solutes. Because if it is freely permeable to these solutes, what's going to happen to these solute concentrations? I saw someone say it. They're going to balance. They're going to equilibrate. And if the solute equilibrates, so it's equal inside and out, what's that kind of situation called then? Isotonic. Now it's in an isotonic environment. And so the water will enter and leave the cell at the same rate. Well, cell survival really depends upon balancing water uptake and loss. I mean, you can't take in too much water, or potentially you can burst. And you can't lose too much water, or you'll become dehydrated. So again, this response of plant and animals to being placed in these various solutions is something that is pretty important to understand. So this shows you typical response of an animal cell and a plant cell placed in hypotonic solutions, isotonic solutions, and hypertonic solutions. Okay? Hypotonic solutions have less solute outside than inside. The water will move from its higher concentration to its lower concentration. And in an animal cell, that will cause cell lysis. Okay? So what would happen if someone were in a bad accident and they were losing blood volume rapidly? If you transfuse them with pure water. Restore their volume, right? Doesn't? Yeah, the red blood cells would burst and lyse because there is solute or molecules inside those red blood cells. The environment would be hypotonic compared to those particular cells, and so the water would rush in. So when we give people saline solution to replace fluids, it's got to be isotonic to the blood cells. Okay? What's going to happen to a plant cell in a hypotonic solution? It'll gain water. And particularly that vacuole will gain water. And it will swell and become turgid, but why won't it burst? Cell wall. Okay, cell wall retains it. And this is actually an ideal situation for a plant because in a turgid state, then that plant tissue has some structure to it, and a leaf, for example, then become erect and have maximum surface area for absorbing sunlight. Isotonic solution, then a red blood cell will have its normal biconcave disc shape. That's the normal type of red blood cell. Plant cells actually become flaccid or wilted in isotonic solutions. There's not this internal pressure developed inside this cell in an isotonic solution. And so there's not that pressure against the cell wall, and the plant cell isn't turgid. So this happens. When this happens, the plant tissue starts to wilt a little bit. And then in a hypertonic solution, which is a very salty solution, the water now moves from inside the cell to outside the cell, and a animal cell shrivels. A plant cell, the cytoplasm actually pulls away from the cell wall, and you can see actual spaces where there is a physical gap between this rigid box and then the membrane of the cell. And that particular process is called plasmolysis. When it used to be legal to use Eladia in Indiana, we would have you do a simple experiment where you would take a little Eladia leaf that you can see through in a microscope, you can see the cells, add a little bit of salt solution to it, and you could actually observe the plant cells shrivel up as the water rushed out. If you then replace that salt solution with fresh water, then you could actually observe these cells reinflate again as water started moving back into the cells again. So pretty important. We talked about this when we talked about cell structures. Here is a paramecium with a vacuole filling up with water. And then that vacuole will fuse with the cell membrane and discharge that water, constantly pumping it out. So this paramecium, if it's constantly having to pump out water, what kind of an environment is it in? Hypotonic or hypertonic? It's in a hypotonic environment. Okay. There's more solute in the cell than outside, so water rushes in. So it's a little bit hard to see. What do these two situations have in common? So here we've got some dead plants over here on a roadside. Here we've got some tomato plants growing in a little flat that also are showing damage and stress. What's this stuff? What's the blue stuff down here in the scoop? It's a fertilizer, okay? Because a lot of people fertilize their plants. So what do these two situations have in common? Why are these plants dying? Why are these plants dying? Commonality. For plants to grow, how do we want water flow to be occurring? Where do we want water to be moving? From the soil into the roots. And if that happens, then the plant cells have plenty of water, they're turgid, plant cells are growing. What would happen if the water were sucked out of the roots? What effect would that have on plant growth? All the plant cells would plasmalize and die. So in both of these situations, plants are dying because the water is leaving the root and going into the soil instead of going from the soil into the root tissue. How come? What governs water movement? The water's leaving the root and going into the soil. Is the soil hypertonic or hypotonic compared to the inside of the root? Hypertonic. Okay. What's causing this soil in this particular situation to be hypertonic? The fertilizer. Anybody know what fertilizer is? It's things like nitrate and phosphate and sulfate. Those are solutes. Salts, if you like. So if you put too much fertilizer on it, you basically suck the water out of the roots and the plant dies. People refer to this sometimes as burning due to over-fertilizing. But really, this is dying. What's happening over here? Why are these plants dying? Why is the soil becoming hypertonic?
Because it's by a road, and in the winter they put salt.
Roadside salt. Okay, you're going to see this in the spring along the interstates and heavily traveled roads that they've salted a lot, where the soil becomes very hypertonic, and consequently those will die. This is just here for your summary. This is kind of a summary of what we're going to be talking about. We're next going to be talking about traffic across membranes. So remember when we mentioned that things move across cell membranes, I said that there were kind of two different scenarios we were going to talk about. And one scenario was movement of smaller molecules across membranes. Okay? And then the other scenario was movement of bigger molecules, or potentially even cells, like a bacterial cell. So let's talk about the small molecule movement first, and talk about some membrane transport phenomena. Okay, passive transport is diffusion across a membrane. Does it require energy? No, the word passive indicates that it doesn't. And what direction do things move according to the principle of diffusion? They go from where to where? Their high concentration to their low concentration. Okay? Simple diffusion occurs when the membrane is freely permeable to the substance. What kind of molecules are the membranes most permeable to? Remember? Nonpolar and non-charged and
Small.
typically small. Okay, so we talked about oxygen, carbon dioxide, good examples of substances that can move through the membrane by this process of simple diffusion. High concentration to low concentration. Facilitated diffusion still involves movement from high concentration to low concentration. And because it's diffusion, it still doesn't require energy. But the difference between this and this is that the membrane is not normally permeable to the substance that's trying to move across. So it needs to be helped. And the molecules that help these substances diffuse across the membrane are sometimes called carrier molecules or transport molecules. And what kind of substances are they? They're proteins, membrane proteins. Okay? There are a couple of different categories of membrane transport proteins. And on the left here is an example of a membrane protein that creates something that we call an ion channel. So let's say these are sodium ions out here. Okay? A lot here, not many here. So the natural tendency, according to diffusion, is for these to move from here to here. But is this membrane normally permeable to sodium ions? No. This is a hydrophobic interior here, and those charged molecules just don't want to be there. Well, these ion channels basically have an interior, the way the protein folds up, so that the inside of this is a hydrophilic channel. Now those ions are okay. They're fine with being associated with that particular hydrophilic environment. And consequently, in the presence of these channels, then these can move from here to here according to the law of diffusion. Okay? Here's another situation that I think I might have a better slide of in a second. Okay, so here's our ion channel again. Here's a different scenario. So we've got a substance out here right now that is higher concentration outside than inside. Natural tendency would be to move into the cell, but these molecules, for some reason or another, can't cross the lipid bilayer. But now we've got a carrier protein. And that carrier protein can bind with these molecules very specifically. You all familiar with the concept of the specificity of an enzyme and its substrate? How only certain enzymes can bind to certain substrates? These carrier molecules are oftentimes like that. They have very high specificity for things. These carrier molecules can exist in two different states or two different shapes. So, might represent it like this, that's one shape, and like this, that's another. And so these carrier proteins can alternate between these two different shapes or conformations. That's just a fancy word for shape. Okay? So what's going to determine whether it's in this state or this state? Whether this molecule binds to it. Okay? Remember that I mentioned earlier on that when we phosphorylate proteins at a phosphate group, that can change their shape. So in this particular situation, the carrier is kind of open, facing the environment. These molecules bind to this binding site, and when that binding occurs, that causes a shape change to this particular carrier. Now it opens the other way. now it's released into the cytoplasm. What happens when this releases from its binding site? It flips back to this state again and can pick up another one. So these molecules swivel back and forth between two different state changes, and as a result, facilitate the movement of that particular molecule through the membrane. Is this requiring energy? No. It is just an aided or facilitated form of diffusion. Okay. This one gets a little complicated, so pay attention. There is another situation that we need to talk about that is really fundamentally important, called active transport. In active transport, what we're doing is, instead of things moving from areas of high to low concentration, which we oftentimes represent as going downhill, in this particular situation, things are moving from a low concentration to a higher concentration, uphill. Okay? So think of that rock sitting up at the top of the hill. It has a natural tendency to roll down on its own, but that particular rock, you're going to need some energy to push it uphill to its higher energy level. Okay? So it's moving against the concentration gradient. And then furthermore, the membrane is normally impermeable to the substances that are trying to move across. So sodium ions or potassium ions can be actively transported. There are several scenarios, one of which we'll talk about in detail, where it's critically important for the function of a cell to have a different concentration of one ion on one side of the membrane than the other. In nerve cells, the two big players are sodium and potassium. How do we maintain that particular situation? Because one of the things that happens when nerve cells are stimulated is sodium ions rush from the outside to the inside, and potassium go from the inside to the outside. That's actually what causes the membrane, the neuron, to polarize and the impulse to be transmitted. But then for that nerve to fire again, what has to happen? We've got to reestablish the gradient again. We've got to get the sodium back out here and the potassium back in here. How do we do that? We do that through these pumps, these active transport pumps, like this sodium-potassium pump that we'll talk about in a second. Plants sometimes actively transport solutes into their cells. What's the advantage of that? Makes them hypertonic. And then water comes in. And they swell and become turgid. Another ion pump that we're going to talk about a lot later on is a proton pump. And cell membranes have to be able to pump protons across the membrane to set up a energy gradient that we can use to make ATP. Well, again, once the ATP flows through the protein to make ATP, how do we reestablish that gradient again? If I'm a proton and I go through this mitochondrial membrane, and that helps to make ATP, how do I get back out here again? We want that process to keep going. A pump is involved, a proton pump is involved. One of the most important things to understand about this is that these pumps work as a result of conformational changes, shape changes. So these pumps are protein molecules that can exist in different shapes depending upon what's bound to them. What's also interesting about these pumps is that some of them are also enzymes. And in the case of the pump we're going to talk about in just a second, the sodium, potassium, ATPase. What does that ACE indicate? Enzyme, okay. Most enzymes end in ACE. What does this sodium potassium ATPase do? It catalyzes the breakdown of ATP. But only when it's active. And sometimes that ATPase is active, and sometimes it isn't. What's that depend upon? Its shape. Okay. This is the sodium potassium ATPase. I know this looks a little bit confusing, but if you go through this kind of systematically, it really does make sense. Okay. The purpose of this pump is to reestablish the situation where there's a lot of sodium outside the cell and a lot of potassium inside the cell. And how does it do that? For every cycle of this pump working, we pump 3 sodiums out of the cell and 2 potassiums inside. So that's going to be the net result of the function of this particular pump. Cell membranes, particularly of neurons, are loaded with these sodium-potassium pumps. Okay. So, here is this protein. This protein can exist in 2 different shapes. One where it's basically facing open to the cytoplasm, and the other where it's basically facing open to the environment. Kind of like that previous example I showed you. But this protein is also an enzyme. And this particular enzyme catalyzes the breakdown of ATP. But the enzyme only works when the pump has a certain shape. It has 3 binding sites for sodium, and it has 2 binding sites for potassium. Okay, so let's start right here. Let's start with this particular starting point in the cycle right now. This is showing when we have the 3rd sodium just binding to this particular 3rd sodium binding site. These potassium binding sites right now are not the right shape to bind those potassium ions. So these sites accept sodium, but these sites are not ready to accept potassium. When the 3rd sodium binds, it changes the shape of that protein a little bit. And now it's an enzyme. And that enzyme can now catalyze the hydrolysis of ATP. And the phosphate group that's released binds to a binding site on this protein and phosphorylates it. What's that going to do? It's going to change its shape. How does it change? It swivels the other way. That's a big change. Okay? But also, it changes the shape of these sodium binding sites. Compare this shape to this shape. It's different. So now the sodiums, which were bound here, they were originally in the cytoplasm, are released out here. 3 sodiums out. When these binding sites change, that causes another phenomenon to occur, where now these potassium sites change shape, so now they can bind potassium. The binding of the potassium causes another shape change. What does that do? That causes this protein to dephosphorylate. And what is losing the phosphate group? It causes the protein to change shape again. So now it swivels back inward again. And additionally, these sodium binding sites change, and so do the potassium sites change. So now the potassium can no longer attach to these, and so it's released into the cytoplasm. So there's a series of sequential changes in the shape of that protein. One thing causing a shape change, causing another, that ultimately accomplishes the goal of putting sodium back out here again and potassium in here. And if this is a neuron, once this has been reestablished, sodium out, potassium in, that neuron's ready to fire again. And part of the firing process involves sodium ions going through a channel protein, rushing in here, potassium's rushing out here, and that propagates a nerve impulse. And then that nerve's got to reestablish and get everything reestablished again. It's got to pump the sodium back out, pump the potassium back in again so it can fire again. This is an example where setting a diagram can really make a lot of sense and give you a lot of help in trying to come to grips with this particular information. I can pretty much guarantee you there will be a question on the exam about the sodium-potassium pump. And probably the easiest way you can study for this and master it is just sit down and reason through what's happening at each of these steps. Okay? So if we start here, why are the sodium ions binding? Because the shape of this binding site is proper for it to bind. Why are potassiums binding? Because the shape of their binding site isn't correct. They don't match. What's the consequence of all three of these being filled up? A change in the protein. So it's now an enzyme. What happens when that's an enzyme? It hydrolyzes ATP. What's the consequence? A phosphate group is available to phosphorylate it. What does that mean? Change of shape. Sodium ion binding sites are different now, released. Potassium ion binding sites are different. They can bind potassium. Swivels to the outside. When the potassiums bind, that causes dephosphorylation. That changes shape again. Pivots inward. Now we're back to our original sodium sites. They can accept sodium. Potassium sites no longer can bind potassium. We rotate through these different shape changes sequentially, and it's all powered by, ultimately, ATP. Now again, ATP, remember, is not like gasoline. It's not powering these pumps like a motor-powered pump would be powered with gasoline. It's driven by the phosphorylation of these particular proteins and the dephosphorylation of those proteins, which facilitates binding of sodium, release of sodium, binding of potassium, release of potassium, and the swiveling of this protein so it opens in and out, in and out. Your cells would not function without this pump, neuron cells. And you are what you are because of your nervous system. Okay? That determines behavior, thought, everything. So at the most fundamental level, all of this little action of this pump helps to determine you as individuals, who you are, and what you can do. This is just another representation of the very same thing, showing you it not in a circular fashion, but in a linear fashion. Some students like this better. Doesn't matter, still conveys the same information. I mentioned proton pumps. Proton pumps are going to be really, really important when we talk about cell respiration. Proton pumps involve moving protons from low concentration to high concentration. Again, ATP is required to make this happen, causing some kind of conformational change in this membrane protein. But once the protons accumulate here, they represent potential energy to do work. And if there are channel proteins through which these protons can move back through again, then we can accomplish some work, just like the water flowing through a dam. But if we allow these to equilibrate, we stop losing the ability to do work. So So we have to keep pumping the protons back out here so they can flow back in here again. That's a big part of what we'll talk about when we talk about cell respiration. In cotransport, there is a membrane protein that actually couples the transport of one solute to another one. And probably one of the most understood examples of cotransport occurs in plants, where we couple the transport of sucrose in plant cells to the active transport of protons out of the plant cell. Show you how that works. So the net result of this cotransporter is that we pump sucrose into the cytoplasm of the cell. What kind of a sugar is sucrose? Remember from lab? Monosaccharide or disaccharide? Disaccharide. It's made out of glucose and of fructose. And it is the principal way in which plant cells store carbohydrate, fat and starch. They make the glucose using photosynthesis. You can chemically modify the glucose using an enzyme into fructose, and then those can be joined together to make sucrose. But eventually this stuff is going to be accumulated in plant cells for storage. So we're moving the sucrose from a low concentration to a high concentration. That requires energy. The energy is actually provided by a proton pump, which pumps protons out to a higher concentration. And then as they move through this cotransporter back to their lower concentration, it kind of drags the sucrose along with it. Okay? So it's actually this movement of the proton that provides the power, if you like, for the sucrose to be transported in along with it into the cytoplasm. That's cotransport. Okay, so now big molecules. What about things like, carbohydrates or lipids, or even a bacterial cell? The bulk transport that I mentioned yesterday, or on Tuesday. So now we're talking about exocytosis and endocytosis. Exocytosis moves substances out of the cell. Endocytosis moves them in. We're going to spend most of the time talking about endocytosis. There are three categories of endocytosis: pinocytosis, phagocytosis, and receptor-mediated endocytosis. What's the distinction between those? Okay, here's pinocytosis. So here's the environment, and we've got a lot of these little water-soluble molecules here. Here's the cell membrane, and it just kind of pinches in. And as it pinches in, liquid is trapped, and any of these molecules that were dissolved in liquid are trapped as well. So it's really very random in terms of which of these molecules are brought along with the water and end up inside the cell. There's no selectivity or specificity. Pinocytosis is sometimes called cell drinking. It's a way that the cell brings in large amounts of water. Here is an electron microscope picture of a cell membrane of an endothelial cell. An endothelial cell is the kind of cells that make up the walls of little tiny capillaries. And capillaries are usually just one cell layer thick. So this would be the inside of the capillary cell. Here would be the membrane, and then this would be the environment. So see all these little pinocytotic vesicles that are forming in here? And this is a way to get large amounts of water and dissolved solute into the cytoplasm. Phagocytosis is called cell eating. And this brings in larger, non-soluble things. Okay, so here's our extracellular fluid again. And here's some kind of food particle, or maybe a bacterium. Cell membrane pinches in, and now that substance is
enclosed within a vacuole. This shows you a white blood cell, no, amoeba, sorry, engulfing a bacterium by this process of phagocytosis. Here's our bacterial cell right here. Here's the membrane pinching in here. And eventually this will be surrounded by a vacuole. And eventually that vacuole will attach to a lysosome where it then exposes the bacteria to all these digestive enzymes, and eventually that cell will be destroyed. That's an important part of your defense system. Yeah?
I'm just not understanding the difference because the membrane pinches in either way.
They're both endocytosis, yes. The difference is that in this particular situation, we're talking about kind of large, chunky things that are not soluble in water. And in pinocytosis, we're talking about things that are water soluble, that are smaller and actually dissolved in the liquid that's being brought in. So here we're bringing in primarily liquid with some dissolved stuff. Here we're bringing in mostly large molecules. Yeah, there's going to be some liquid in here too, but it's mainly big food. Okay?
For both of them, is it just like a defense system in your immune?
Really, it's the phagocytosis that is the defense system part. Yep.
So the pinocytosis isn't destroying the liquid, or the little...
Yeah, the pinocytosis is just kind of a way to bring in large amounts of water and solute.
Okay.
It's not defensive. The third one is a phenomena called receptor-mediated endocytosis. So what's going on right here? Within the membrane of these cells are some specific receptors that will bind molecules very specifically. So these little red Ys right here represent a membrane receptor. And the membrane receptor is specific for these purple triangle molecules. It won't attach to the green circles. Okay? So when these vesicles form, purple triangles tend to bind to these particular receptors and be held there. Green circles, they might diffuse out, they might stay in there. So there's going to be a greater likelihood that you're going to get these purple triangles into this vacuole than you will the little green circles, because there's receptors for them. So this is a way in which the cell can bring in some very specific molecules and enhance the chances that those will get into the cytoplasm. And this just shows you a couple of electron micrographs here. You can see the pinching in of the vesicle right here. And these are molecules that have been attached to the receptors. So this eventually will become this vacuole right here. Well, there's a situation physiologically that is very related to receptor-mediated endocytosis. This particular situation is called familial hypercholesterolemia. Kind of a mouthful. Any idea what familial refers to or indicates about this condition? Genetic. Okay, it's an inherited disease. And hypercholesterolemia, what's happening to your cholesterol levels in this condition? Really elevated. Okay? If you suffer from this particular condition, your cholesterol levels can be up in the 500s or more. Okay, and that's way higher than it should be. And higher cholesterol levels do have associated risks for coronary artery disease, etc. Okay, so it's a bad situation. In fact, people that suffer from this typically die from coronary artery disease in their, 30s and 40s. That's pretty serious. There are different kinds of cholesterol, and they actually associate with proteins. So we've got lipoproteins, and these circulate around in the blood. And there's two types of lipoproteins, high-density lipoprotein and low-density lipoprotein. Which is worse for you? HDL or LDL? Low-density lipoproteins are more damaging to your health. Well, in the membrane of normal individuals are all of these receptors that bind LDL. and they can bring the LDL from the blood into the cell and store that lipid because of receptor-mediated endocytosis.
Okay.
So, if we have a situation where we've got genes that code for these receptor proteins, and let's say that capital L is the normal allele, that codes for the good functional LDL receptor. And lowercase l is the mutant allele. So what's going to be your situation if you are homozygous? You get one copy of that from each parent, one good copy of the gene. Then you will have a normal number of receptors. You will have the ability to remove this LDL from the blood serum and store it in cells, and your serum cholesterol level won't be too high. What if you got one from each? Well, then you'll have some ability to remove cholesterol, but your cholesterol level will probably be elevated. Now, this isn't the only thing that contributes to high cholesterol, but this can be one of the things. And then if you lose the genetic lottery, and you have 2 bad copies of that gene, then you make very little of any functional receptor protein, and so now you can't remove that cholesterol from the blood and store it in cells, and you have highly elevated cholesterol levels. Okay? So again, a medical condition associated with a particular genetic explanation having to do with a membrane protein. Okay, on Tuesday, we will finish up membranes and talk about membrane structure and how it relates to function. And that will be the ending material for exam 1. So that way you're going to have a week hiatus where we're not kind of cramming new material in right up to the exam. But we will start with new information on Thursday, and that information will be on your second exam. Now, for Monday, please remember to have read the lab, the cell membrane lab. Because the cell membrane lab is going to be a direct application of what we've just talked about. We're going to talk about osmosis and diffusion, membrane transport. So you're going to help yourself if you know what you're doing. Okay? Next week, Thursday and Friday, we will cover new material for exam 2. We'll just talk about it in class. A week from this Monday, Dr. Morden will be here working with you on the kind of lab stuff. And then if you have exam questions, he will leave the remaining time to do a little bit of review. And then Tuesday, you'll have the exam. I'm going to be out of town Monday and coming back Tuesday. And my intent is when I get back, I will have Wednesday to grade the exam then. And hopefully I'll have it back to you then on Thursday when you come to class. Okay? So, quiz Monday, read your lab for Monday. Stay warm. Have a good weekend.